Hello friends, again welcome to my channel. Now today we are going to discuss about another RNA molecule, that is the transfer RNA molecule, their structure and the function. In the previous lecture we are already discussing about the structure and function of the ribosomes. So now we are going to discuss about the structure and function of this transfer RNA molecule. Now this transfer RNA or the tRNA molecule, which is a type of the RNA molecule, it is responsible for the decoding of the codons which are present on the mRNA molecule during the translation process. Means for the protein synthesis procedure, majorly the information which is present within the mRNA molecule in the form of codons, it is get decoded into the form of sequence of amino acid, and this tRNA molecule it is help for this decoding procedure. This tRNA molecule it is acts as an adaptor or the carrier of the amino acid, which is responsible for transferring the corrected amino acid within the growing polypeptide chain during the translation process. The tRNA molecule is containing only about 70 to 90 nucleotides within the length, as these are smaller in size. Means whenever we are comparing the three different types of RNA molecule, that is the messenger RNA, transfer RNA and the ribosomal RNA, out of these three types of the RNA molecules, the tRNA molecule, these are the smaller RNA molecules. The molecular weight of this tRNA molecule it is ranges in between 25,000 to 30,000 Dalton. These tRNA molecules these are encoded by the DNA within the cell nucleus, and majorly these are transcribed with the help of RNA polymerase III. Now we are switch over to the what are the specific properties of this transfer RNA molecule. This transfer RNA molecule can make the hydrogen bonds with the messenger RNA and also can form the ester linkage with the amino acid. Means during the amino acylation of tRNA molecule or the charging of tRNA molecule, this ester linkage these are get formed with the amino acid. The part of tRNA that binds with the codon of mRNA, it is called as the anticodon loop of tRNA molecule. This tRNA it is a single stranded RNA molecule which has the proximity to fold upon itself and creates an intra-complementary base pairing, which is responsible for giving and stem and loop structures which is hydrogen bonded. The tRNA molecules these are also contains the modified unusual bases, means no doubt the common nitrogen bases these are also present within this tRNA molecule like the other RNA molecules, that is the uracil, adenine, guanine, cytosine. These normal nitrogen bases these must be present within the transfer RNA molecule, but along with these normal base pairs there is also presence of some of the unusual bases, for example the inosine, then the pseudouracil, then the dihydrouridine. All of these unusual bases these are also present within the transfer RNA molecule. These present modified unusual bases and the intramolecular base pairing of tRNA make it stable and protect it from the degradation. Now majorly the tRNA molecules these are appear within the three different form structures, that is the primary, secondary and the tertiary structure. So firstly we are focusing on what is the primary structure of the tRNA molecule. Now within this primary structure of tRNA molecule, it is considered to be only a linear single stranded ribonucleotides which are running within the 5' to 3' direction. The number of nucleotides these are ranges in between 60 to 90 nucleotides, and the most commonly these are the 75 or the up to the 76 ribonucleotides. Based on these modified bases as we are discussed earlier, the linear sequence of tRNA molecule it can be divided into the three arms. Means on the basis of these unusual bases, that is the D arm. This D arm it contains the modified base dihydrouridine. The anticodon arm containing the unusual base like that of the inosine, which is derived from the adenine, or the pseudouridine which is derived from the uracil, or the lysidine which is derived from the cytosine. This lysidine it is present within the T-C arm of the tRNA molecule. which majorly along with that it also contains the pseudouridine. Now in addition to these three arms with the modified bases, an extra arm it is present at the 3' terminal, usually containing the CCA nucleotide at its 3' end, and it is referred to as an acceptor stem, or it is called as an acceptor arm, and majorly it is the site where there is the attachment of specific amino acid to the tRNA molecule during the amino acylation of tRNA molecules, and which is an initial step during the translation process. This is on the basis of the anticodon which are present within the anticodon loop of the tRNA molecule. There is the attachment of the specific amino acid. As we say that these tRNA molecules, these are acts as an adaptor molecule or carrier molecule of amino acid. So majorly this 3' end of the tRNA molecule, it gives the binding site for the amino acid. The D arm serves as an recognition site for the enzyme aminoacyl tRNA synthetase, which is applicable during the amino acylation of tRNA molecule. The anticodon arm, it binds with the codon of the mRNA molecule, that is during the decoding of the codons on the mRNA molecule there is the use of this anticodon loop. The T-RNA arm, these are containing the ribosome binding site, that is during the translation process whenever there is the binding of this tRNA molecule with the ribosomes, for that purpose this T-RNA arm, these are helpful. Now we are switch over to the secondary structure, which is also known as an clover-leaf structure or the clover-leaf model of the tRNA. Now this secondary structure of tRNA, it contains up to the 74 to 95 nucleotides. These are stable and fold on itself to give a clover-leaf like appearance. The 4 base paired arms, these are called as acceptor arm, D arm, T-RNA arm and the anticodon arm. Now the acceptor arm, it contains about 7 to 9 base pair in length, and this arm, it is formed by the base pairing of 5' terminal and the 3' terminal nucleotides. 5' and 3' terminal nucleotides. The carboxylic group of amino acids, these are joins with the free 3' hydroxyl group of adenine within the CCA, which is a terminal nucleotide present on the acceptor arm to the aminoacyl tRNA. The acceptor arm, it is located opposite to that of the anticodon arm. Again the D arm, which is the part of the tRNA molecule, it is consist of a stem that is about 4 to 5 base pairs in length and which end in a loop that is usually contains the modified base, that is the modifier pyrimidine base, that is the dihydrouridine. So the name it is the D arm on the basis of the presence of the unusual modified base. The D arm, it is serves as an recognition site for the enzyme aminoacyl tRNA synthetase. Now the anticodon arm, which is again present opposite to that of the acceptor stem, it is containing about the 6 base pair in length and which ends in a loop due to the 7 unpaired bases, out of which the 3 bases form an anticodon. 3 bases, they are form an anticodon. And these anticodons which are present within the tRNA molecule, these are responsible for the decoding of the codons on the mRNA molecules. The T-RNA arm consists of a stem that is 5 base pairs long and which ends in a loop that is further contains 7 unpaired base pair, usually containing the thymine, pseudouridine and the cytosine, and due to that the name it is the T-RNA arm. And these are possessing the binding site for the ribosomes. Again, one of the important structure which is in the tRNA molecule, it is the variable arm. These are vary within the length from 3 to 21 nucleotide depending on the type of amino acid they code. The variable arm, it lies in between the anticodon and the T-RNA arm. The variable arm, it accounts for the stability of the tRNA molecule. Now the third one, it is the tertiary structure of the tRNA molecule. Now majorly this tertiary structure, it is a 3-dimensional structure of the tRNA molecule and majorly they possess the L-shaped structure. This L-shaped structure or the tertiary structure, it is resulted whenever the stem of the acceptor and the T-RNA arm stack at the one side. There is the stacking of these two arms, that is the acceptor and the T-RNA arm on one side, and the stem of Anticodon and the D arm, these are also stacked on the another side. Means there is the stacking of both of these arms on the one side. Then these are responsible for forming the extended helices. Now both of these extended helices align at the right angle, and in doing so the D loop and the T side C loop, these are aligned together. These are aligned together. This tertiary structure, it is the most stable structure of the tRNA molecule, and its stability it is acquired due to the hydrogen bonding in between the nitrogenous bases and also in between the nitrogen base and the ribose phosphate backbone. To synthesize the proteins by the reading codon in a mRNA, a tRNA molecule must have the tertiary structure. Means whenever they are present within this tertiary structure, then and then only they are responsible for the decoding of the codons which are present on mRNA molecule during the translation process. Here within this figure you can observe the 2-dimensional, 3-dimensional and the simplified structure of the tRNA molecule. That is, this 2-dimensional, it means it is a secondary structure which contains the T side C loop, then the amino acid acceptor site, that is the acceptor stem, then opposite to that the anticodon loop it is present, then in between these two loops there is the variable loop it is present. The similar structure it is observed in case of the 3-dimensional structure, that is the L-shaped structure. These two arms, these are stacked on one another. Due to that these are formed this L shape, and this L shape it is essential for the decoding of the codons during the translation process. Now the last point within this, it is the function of tRNA molecule. What are the specific functions which are done by these tRNA molecules? Now majorly these tRNA molecules, these are the vital molecules which are taking part within the protein synthesis procedure and majorly transport the amino acid to the ribosomes, where these amino acids are added according to the codon sequence, that is the codons which are present on the mRNA molecules. These are also participated within the non-protein synthetic processes, that is the such as the primer during the reverse transcription of retroviruses. These ribosomes, these are acts as an primers during the reverse transcription of retroviruses. It is also known as an adaptor molecule because as these are giving the site for the attachment of the amino acid and responsible for transferring this amino acid within the growing polypeptide chain during the translation process. The mutation within the tRNA or the tRNA processing are linked with the various human diseases like that of the cancer and the neurodegenerative diseases. Majorly in the recent year, the tRNA molecules, these are altered to the accommodate desired anticodons that result within the translation of individual amino acid and to do some specific function. Now majorly these tRNA molecules, due to the alteration within these tRNA molecules, these are participated within the development of the resistance within the bacterial strains against different antibiotics, and again there is also improvement within the pathogenicity of these microbial strains. So these are the different functions which are done by the tRNA molecules. So if you like this video, please subscribe my channel and also share this video with your friends. Thank you.